In a breakthrough study with promising implications for how we understand and potentially treat age-related obesity, scientists at City of Hope have discovered a new type of adult stem cell that appears to be a driving force behind the increase in belly fat as we grow older. This discovery sheds new light on the mechanics of aging and offers a genuinely hopeful outlook on how science might one day help us maintain healthier body composition well into later life. The research, published in Science, zeroes in on a group of cells called CPS, short for committed preadipocytes, age-specific, which seem to emerge around middle age and are particularly efficient at transforming into fat cells. These CPS are triggered by a signaling pathway known as LIFR, leukemia inhibitory factor receptor, which acts as a kind of molecular switch for abdominal fat production in older bodies. What's truly remarkable here is that these cells don't just contribute to fat storage. They actively produce new fat cells at a much higher rate than their younger counterparts. That means the challenge of belly fat in aging adults isn't just a matter of metabolism slowing down or exercise declining. It's a result of biological programming that actually enhances fat cell production. This discovery doesn't merely explain a frustrating reality. It offers an opening to intervene. If LIFR is the trigger for CPS to proliferate, then blocking or modulating this signal could, in theory, prevent the chain reaction that leads to age-related belly fat. The team's work was rigorous, beginning with preclinical trials in mice and later validating their findings using human tissue. They found that older mice had significantly more active APCs, adipocyte progenitor cells, the parent cells that eventually form fat. And when these older APCs were transplanted into young mice, they still produced large amounts of fat. This means the fat-producing behavior is hardwired into the cells themselves, not simply a byproduct of aging physiology. Another extraordinary feature of this research is its clear implication that aging doesn't just break things down in the body. Contrary to the assumption that stem cells decline in activity as we age, these CPS seem to increase in function, at least in their ability to generate fat. This is a rare example of aging enhancing a biological process, even if it's one that most of us would rather avoid. In that sense, the research challenges some outdated notions about aging as a purely degenerative process. Instead, it suggests a more complex picture where some cellular systems rev up rather than slow down, and that nuance offers important leverage points for medical science. Beyond the immediate focus on fat and metabolism, this discovery fits into a broader and more optimistic narrative about how modern medicine is starting to decode aging at a molecular level. We're beginning to see aging less as an unstoppable slide into decline and more as a puzzle with specific, actionable pieces. The fact that these stem cells and the LIFR pathway that activates them can now be identified and studied in detail means that future therapies could be precisely targeted. Whether that leads to a drug, a gene therapy, or another approach altogether, it's now possible to imagine interventions that reduce harmful fat buildup without compromising other aspects of metabolic health. Importantly, the research team also found similar stem cells in human tissue, which bolsters confidence that these findings aren't just an animal-specific phenomenon. By applying single-cell RNA sequencing to human fat samples from people of varying ages, they confirmed that CPS increase in number as people reach middle age. That means this isn't just a fascinating scientific observation. It's a real phenomenon affecting countless people and contributing to the global rise in age-related metabolic disorders. The ultimate goal now is to understand these CPA cells even better, how they form, how long they persist, what else they might affect, and then to find ways to block or remove them before they lead to serious health problems. Researchers are planning to track these cells more closely in both animal models and human subjects, and to begin developing treatments that could one day make age-related belly fat less of an inevitability and more of a manageable condition. In a world where aging often comes with a sense of inevitability and loss of control, this discovery offers a welcome shift in tone. It suggests that the future of aging could look very different from the past. Not because we'll stop aging, but because we'll finally understand it well enough to make better choices and live healthier, longer lives. It's not about vanity or we're trying to hold on to youth forever. It's about harnessing the power of biology to age with vitality, energy, and dignity. That's a future worth working toward.